Welcome back, botanist. Let's see, if you haven't done it already, make sure you grab your lab notebook, your colored pencils and pens, and get ready. All right, so we've been talking about cells, then we talked about cell types, our enchemas, and then we talked about tissues, groups of cells performing a similar function. Now I want to shift your focus on how one cell can grow and become many. All of you started out as just a single cell. So did any organism that was the product of sexual reproduction. Imagine the tallest redwood tree in the forest. If it came from a seed, it was once one cell. That one cell divided many, many times to develop into the complex multicellular towering tree you see above. So let me start screen sharing. And we'll start our notes. So if we think about a cell, there's two stages in the life of a cell. Now, most of a cell's life is going to be spent in what we call interphase. And in interphase, a cell is doing whatever its normal function is. So maybe it's a photosynthetic cell, right? And it's photosynthesizing. Maybe it's a storage cell, storing up starch in the form of those leucoplasts. It's doing whatever its normal function is. It's gonna enlarge, right? Get bigger, right? Just that one cell, just enlarging, getting bigger. It's gonna synthesize DNA and proteins. Actually, I meant to say proteins and organelles. Sorry about that. So it's going to synthesize proteins and organelles. And the DNA, before it can enter the next phase, is going to have to be duplicated. The DNA is going to have to be copied before we can enter the second phase in the life of the cell. And the second phase is called mitosis and cytokinesis. And it's through these processes, mitosis and cytokinesis, that one cell can divide and make two. Now, mitosis is going to divide up the DNA. Right? That's how our genetic information is going to get separated. Cytokinesis is actually going to divide up the cytoplasm. And whatever's inside that cytoplasm. So if there's organelles in there, they're going to get divided up between the two cells that are produced. Now, before we start looking at these processes in more depth, I want to make some drawing of what we call chromosomes. Now, when you see this word chromosome, it just means the condensed form of DNA. Now, our DNA is going to have to condense. It's going to have to tightly wind up in order for it to be divided. Now, before it can be divided, right, our DNA is going to be duplicated. And so I'm going to draw what we call a duplicated chromosome. And all I mean by that is that it's already been copied. I'm going to make my particular chromosome red, not for any particular reason, just because. And we'll look at some real pictures of some in a second. Right, all of this is my duplicated chromosome. Right, this whole thing is that duplicated chromosome. The two halves of it are going to be attached here in this little central region. And if I draw kind of an imaginary line down the center, I'm dividing up that duplicated chromosome into what we call sister chromatids. And so this half is a sister chromatid. And this half is a sister chromatid. These two are sisters to each other because they're actually going to be identical copies of each other. And so it's along these chromosomes where we're going to find our genes. And genes are just sections of DNA that code for proteins. So I'm just going to make this a flower color gene. If this sister chromatid has a copy of the flower color gene right here, we're going to find an identical copy of it over here on this sister chromatid in the same location. Our sister chromatids are going to be identical to each other. They're going to have the same genes, the same versions of those genes, and the same locations. The last thing that I want to add to this drawing is just what we call this little central region that connects our sister chromatids. We call this the centromere. So that centromere just connects the two halves of our sister chromatid. Now let's look at this really quickly to see some of these in real life. So this is just a picture showing you those two parts in the life of a cell. Most of this you can see is interphase. There's different phases within that. I'm not worried about those distinctions. In some other courses, you might be worried about those. It's just all interphase for me. Down here, we see how our cells actually divide, right? We see mitosis and cytokinesis. And ultimately, that's going to produce two separate yet identical cells. Now, here we see a duplicated chromosome. And this is a real one. So you can see why we kind of draw them as these cute little X's, right? All of this is one duplicated chromosome. That centromere would be right here. And each one of these halves, remember, is a sister chromatid. Well, we're going to see that ultimately, during mitosis, our chromosomes are going to be in this duplicated form. Our sister chromatids are going to get separated from each other, and ultimately they're going to get separated and put into separate cells. Well, now what I'd like to do is I'd like to illustrate or model for you what this all looks like, right? How our cells are going to divide. So we're going to be going through mitosis. We're going to be seeing that ultimately what happens in mitosis is that we're going to get one parent cell dividing to produce two identical daughter cells. And as far as how mitosis functions, right, overall in an organism, well, it functions in growth, right? New cells are going to be produced by mitosis. They can then mature into things like tissues. It's also going to function in repair and replacement. Of cells that get damaged or worn out within reason. And if it's an organism that's capable of it, mitosis is also going to be how asexual reproduction can occur. Now, humans aren't so good at this, but plants are, fungi are, right? They're going to use mitosis to asexually divide and produce copies of itself. Now, in just a minute, we're going to be drawing our way through the phases of mitosis. And we're going to see that there's 4 phases. And I remember the acronym PMAT to help me remember the phases in order. And in just a few moments, we're going to be drawing our way through just what each one of these stands for and what it looks like. Now, we're going to start making some drawings. And this first cell isn't actually going to be in mitosis. It's going to be in interphase when a cell is where a cell is most of the time. And I'm going to make these drawings where I'm going to have essentially this little box. This box is going to be my cell. And I'm just going to use a single line to represent that cell wall. Because you know, all cells, all plant cells are going to have a cell wall and the plasma membrane. I'm just going to draw it as a single line. That's going to be my cell wall and my plasma membrane. That means that if I'm just inside of that little line there, I'm inside the cytoplasm. And how we actually identify a cell that's an interphase is we're going to see that's going to have well-developed nuclei, or a nucleus singular. So I'm going to draw my nucleus over here. My DNA is going to be isolated from the cytoplasm by that nuclear envelope, that edge on the outside. And as far as what it looks like, we'll think of that very basic cell we saw on something like the red onion or the drawings we made. Our DNA is going to be in the form of chromatin. It's not going to be wound up into those visible chromosomes. And within that nucleus, we're going to find one or more of those nucleoli, or nucleolus singular. So this is what a cell in interphase would look like, right? This is going to be when it's doing its normal function, right? Well-developed nucleus, DNA in the form of chromatin not wound up, and one or more of those little nucleoli within the cytoplasm. Well, this is going to set the stage for mitosis and cytokinesis. And before a cell can enter into them, remember that DNA is going to have to get duplicated or copied. And so that's going to have to occur before we can enter into mitosis and go through PMAT, right? These stages of mitosis. Well, let's start doing that. So the P in PMAT stands for prophase. And this is going to be our first stage in mitosis. And once again, I'm going to draw this box. Remember, this is just going to represent my cell wall and my cell membrane. I'm not going to label it on every picture, but that's just going to contain. Now, some important things are going to happen during prophase. One of the things that's going to happen is, remember, mitosis ultimately is going to divide up our DNA. So our nuclear envelope, right, the outer edge of that nucleus that isolates the DNA from the cytoplasm, is going to start to dissipate or break apart. So I'm going to draw it as this kind of dashed line right here. That's just that nuclear envelope breaking apart. It's literally disassembling itself. It's disassembling itself so that ultimately our DNA can get divided. While our DNA, our genetic information, is going through changes too. The most dramatic one is that our DNA is going from the form of chromatin to those duplicated chromosomes. And so I'm just going to draw this kind of like a little bit shaggy like that. I'm going to draw two chromosomes, one in red and one in blue, just to kind of help me keep track of them, not for any particular reason. These are my duplicated chromosomes that are in the process of condensing, or in the process of winding up. So those duplicated chromosomes are going to be condensing during prophase. And there's one final thing that's going to happen during prophase, and that's something called spindle is going to start to form. And I'm just going to illustrate it kind of like this. The spindle is going to be forming in the cytoplasm because we're going to see that it's actually going to attach to those duplicated chromosomes, and ultimately it's going to be responsible for ripping them in half. Well, this is going to set the stage for the next stage of mitosis, the M, metaphase. And just include that box again. Remember, that's my cell wall and cell membrane. And I remember metaphase middle, because what happens is our duplicated chromosomes are going to be fully condensed, fully wound up, and they're going to line up in the middle of the cell. So I'm going to have my blue one lining up there. I'm going to have my red one lining up here. I'm just drawing 2 chromosomes for simplicity's sake. There'd be more than that in real life. And what causes them to line up in the middle of the cell is that the spindle has attached to them and pulled them into position at that centromere region. So that's still the spindle right there, right? It's attached to those duplicated chromosomes and it's literally moved them into position. And this little sort of imaginary line where they all line up, we call the equatorial plane. And you can think of the equator, right? The equator runs through the middle of the Earth. This equatorial plane runs through the middle of the cell, and it's where those duplicated chromosomes line up during metaphase. Well, metaphase is going to set the stage for our next stage of mitosis, and that's anaphase. In anaphase, our duplicated chromosomes are going to be ripped apart from each other. So I remember anaphase apart. Here's my little box again. Our duplicated chromosomes are going to get ripped in half. One half of my red one is moving to one pole of the cell. The other half is moving the other direction. One half of my blue is going to move toward one pole. One half is going to move toward the other. And the reason they're moving is they're still attached to that spindle. That spindle has literally pulled them apart at that centromere and is literally pulling them to opposite ends of the cell. Well, our last stage in mitosis is going to be telophase. Here's my little box again. Once again, representing my cell, right? Cell wall and cell membrane, cytoplasm in here. And what's going to happen in telophase is that we're going to get 2 nuclei reforming, right, at opposite ends of the cell. So we're going to get a nucleus reforming right here. And inside each one, we're going to have 1 copy of the red chromosome and 1 copy of the blue chromosome. So this is just our nuclei reforming during telophase. And at this point, we've divided up our DNA, right? It's now getting packaged into these separate yet identical nuclei at opposite ends of the cell. So we divided up our DNA. Now we need to divide up our cytoplasm, right? Actually take this cell with 2 separate nuclei and divide it up into 2 cells. Well, that's the business of cytokinesis, and in plants, this occurs through something called a cell plate forming. And it kind of looks like this little blurry line like that. So the cell plate is going to be a new cell wall and plasma membrane. And eventually, it's going to be complete and go all the way across and separate this into two cells. And that's how cytokinesis happens in plants. Right, that cell plate is going to form and divide this up into two separate yet identical cells. All right, in lab, we'll be seeing this process happen, right? We'll be seeing this in cells that have been sectioned, and we'll get to see all these different stages. Probably show you some time-lapse videos too as well. All right, that's it for now. Don't forget to watch part 2, which is going to be how roots develop.